think about with gene expression is that every cell within an organism has the exact same genome, the same DNA, but then not necessarily the same RNA and the same proteins. So let's think about this like a blueprint. So if you have blueprint, so like DNA coding for how to make an organism, so you'd have a blueprint coding for how to make, say, a house. This is the only copy that you have, right? The original copy. It's not something that you want to jeopardize. If you break it in some way, you're in a lot of trouble. So part of the central dogma is DNA being transcribed. Into RNA, which is your working copy. So the working copy is then something you could take out to the job site to actually use as the blueprint to build your house, while your original copy is safe. And then, of course, you want to actually build your house. And so RNA being copied into DNA through translation actually gets the house made from your working copy while your original is safe in the genome. This extends a little farther, actually, so let's see. My computer's not interested in this today. So let's think about having a blueprint to make a house. You know actually that all of the rooms inside of a house are not the same. Same as all the cells inside your body are not the same. You have heart, you have liver, you have skin, you have hair. So how do you actually take one blueprint and make all of those different things? So you can think about different rooms of the house. There are some things that you need to have in every room. So you probably want to have light in all of the rooms of your house. Maybe you want to have paint or wallpaper on all the walls of your house. But then there are also things that are different, that you want different things in your rooms. So maybe in the bedroom you'd want carpet, whereas in the bathroom you'd want tile. And then in the bathroom, sorry, yeah, there's your tile. And then in the bathroom, specifically, probably the only place in the house that you want a toilet, right? Maybe not so great to have in the kitchen. So the same sort of holds true in our cells, right? You don't want your heart cells and your liver cells and your skin cells and your eye cells to all have the same genes being expressed. So that's sort of the underlying difference between the different types of cells, are different genes being expressed. So some the same, but then some different. So we're going to talk about how you actually go from that same blueprint to getting different sets of genes expressed. So here's an example. Plants have different organs, same as us, and then here's some different cell types. So gene expression, different genes underlying those differences. So let's start by talking about transcription. So that's the process of taking the DNA and going to RNA. So a few things before we can start talking about transcription are the different molecules. So you've seen DNA before. You have the sugar phosphate backbone and the nitrogenous base. RNA is a very, very similar molecule. The sugar phosphate backbone and nitrogenous bases. Do you see the difference here between the two? So deoxyribose and ribose, so this OH group is present on your RNA. Sorry? Okay. But then not on the DNA. So subtle change, but big difference. And then looking at a whole molecule of DNA. DNA we know is double-stranded. We have C's, G's, A's, and T's. C's and G's base pair, A's and T's base pair, and DNA is, generally speaking, a double-stranded molecule. But just that one little change, now we have RNA, which is a single-stranded molecule. Almost the same bases, C, G, and A are all the same, but RNA has a uracil instead of a thymine here. And again, the difference between uracil and thymine is quite subtle. So there's a CH3 on the thymine here that's not on the uracil there. But still, RNA is able to base pair similar to DNA, although generally speaking it is double-stranded. But the same rules apply. C can base pair, but make those hydrogen bonds with G, and A can then make hydrogen bonds and pair with U. So if you remember, one of the themes from the last unit was that small chemical changes make big biological waves. So here's another example of that. Just that change in OH group can result in big changes in the molecule. Especially DNA is a quite stable molecule, and then RNA is relatively speaking unstable.